Hey guys, it's Mrs. Shannon here, and in this video what we're going to do is basic staining of slides, different cell types. We're going to include the cheek cell as well as a plant cell. So when you're at your lab station, you must make sure, especially because we're going to be using cheek cells today, that you have containers for all waste. You should have two different containers, one for any slide that you use that comes in contact with your cheek cells, and one for any of the toothpicks you use. You also need to make sure you have water. You're going to need microscope slides, and there should be a paper plate with it on it. You can see kind of a paper plate here with an onion. You're going to have an onion. You're going to have toothpicks, which I think I just left my toothpicks on the other counter. Let me grab them really quick. And you're going to have these little squares called cover slips, okay? And so I'm going to kind of walk you through how to stain it. When we use a stain, what we're doing is we're using another type of indicator. We talked about indicators helping us identify if something is present when we looked at biomolecules. We talked about Benedict's for our glucose turning orange. We talked about burette for proteins. Well, there's other types of stains too, and these stains that we're going to use stain specific organelles, okay? So we're going to use two different types today. We're going to use methylene blue for our cheek cells, and we're going to use iodine. Now, methylene blue stains for genetic material like DNA, and that's found in your nucleus. And so it's useful to try to find a cell to see if there is a nucleus in a cell. So to do this, it's going to talk to you in step one about making a wet mount. To do a wet mount, okay, and I might kind of fluctuate back and forth with our microscope here. To do a wet mount, what you're going to do is you are going to make sure you have a toothpick, and you're going to want a pipette for the water. So all you're going to have to do is take a toothpick, and please, if you just ate something recently, do not be the person to do this. You're not going to want it because it's going to have like chunks of food material in it. You just want your straight up cheek cells. You're just going to rub it, okay, back and forth, and then we're just going to scrape it on the middle of the slide, and I'm scraping it right here in the middle of the slide. Okay, right away I get rid of my toothpick. Notice I am wearing gloves today, okay, because we are dealing with bodily part fluids, and we also are going to be wearing an apron because we are going to deal with stains. Okay, after you're done with that, you are going to add a drop of water right onto that. This just kind of like dilutes it a little bit and helps spread out the cheek cells. So just add one drop of water here. Okay, you can kind of see it right there. And then what's going to happen is I'm going to put my cover slip on. Now I have to open my cover slips up, so give me a moment here. And we use cover slips because that way we keep the item that we want on the slide. It makes it easier for us to stain, and it also protects the objective here that we're going to be using, the bottom here, the lenses. It protects it from any kind of debris that might get on it. Now, to make a wet mount, once you have your water on your sample, and this goes for anything, not just cheek cells, but any kind of specimen, whether we're doing the onion cell in a few minutes or anything else. To do a wet mount, once you have the water onto your sample, you just take the cover slip. Let me see if I can do this so it's we're going to try it this way. You can see it's at a 45-degree angle, and I just let it drop. Okay? That way, that's the easiest way to get out all air bubbles, but also not smush down your sample because we don't want to break any of our cells. So once you do that, you're going to take your stain. Your stain for the cell here, for the cheek cells, is methylene blue. And all you're going to do, I'm trying to do this sideways. I hope I don't make a mess. Is you're going to just put a drop right by where that cover slip and slide meet. And thanks to our properties here of cohesion, it should be brought right into that slide underneath it. Now what you can do then is go through and you'll want to kind of blot the sides. If you've got some on the sides, I just take paper towel. This stuff will stain. Be careful. Okay, and I just kind of get it up. Okay. Another thing that you can do is when you go to put it on, and I'm just trying to blot it some more, is remember how like capillary action, the paper towel will draw that liquid towards you? If you put paper towel on the other side when you put your cover slip on, it will help draw that across a little bit more. So you can kind of guide where it's going. Okay. Okay, and then once you've got that, you're going to slide it on here, and we're going to use our techniques for locating items on a microscope. Slide, starting at our low one. We're going to use our coarse adjustment and then our fine adjustment, and we're going to try to see if we can find any of the cells. You will eventually get to the highest objective. Make sure you don't use the coarse adjustment then. And then what's going to happen is you will sketch what you see. So you'll sketch it at 4 times, 10 times, 40 times, and this is the other one. This is with the oil microscope. I don't have a spot on here for you to draw. So what I'm going to do is have you just draw on the back of your paper. Okay, just draw circles. Just draw a circle to represent the field of view and draw what you see there. Okay, hope this helps. Stay tuned. Actually, you know what? I'm just going to go ahead through, pause it here so you can do the cheek cell, and then we can go on and we can do the onion cell. Okay, so now hopefully you're ready for the onion cells. Onions are plants, they're eukaryotes. Now I just took my gloves off. I'm going to probably put some back on before I stain it because we're going to use iodine for this one. Iodine, we talked about this in our notes for our biomolecules. It is an indicator of starch, those polysaccharides. And so if starch is present, then it will turn a dark color. Now plants produce a lot of starch. They produce a lot of glucose thanks to photosynthesis. And the organelles that often happens in is chloroplasts. And then they store it in other types of plasts that are also found in it until it is needed. So we're going to see if we can locate them. Now for the onion, you do it the same way. You take your sample of your onion, and I want you to see this sample here. It is not very big, okay? It's like one little bitty layer here. And you're going to lay it on the middle, okay? If we get it too thick, we're not going to be able to get the light through. And then just like before, I'll add my drop of water. I'll add my cover slip, which I'm going to save the cover slips for you. And then I'm going to add my iodine. I do it the same way. I add the iodine to the side of the cover slip. Okay, if I put a paper towel on the other side and this says it in the directions, it will help kind of draw it across underneath that cover slip. Blot as needed, and then begin viewing the onion slide under there. And that's 2 different bacterial slides you can, or not 2 different bacterial, 2 different eukaryotic slides that you can look at and look at different cell types.